All right, my name is Bastian, and I'm a postdoctoral fellow here at the Natural History Museum, and I'm interested in jellyfish evolution, and at the moment in particular in the evolution of life cycles in this group. So when most people think about jellyfish, they have something like this in mind, jellyfish bloom. This is the common moon jellyfish, Aurelia. What most people are not aware of, though, is that there's a second hidden life stage to the jellyfish. So all jellyfish essentially start out, or most jellyfish start out as a polyp. This is a polyp that attaches to the seafloor. In this case, it's growing in a lab on a glass slide, but in nature, they would just attach to rocks or any sort of hard-bottom substrate. The polyp has tentacles similar to a jellyfish with which it catches prey. Every so often, when the conditions are right, though, in the case of Aurelia, for instance, it forms this long string of baby jellyfish at its oral end that are then released and grow up into the adult jellyfish. In other groups of jellyfish, the whole polyp may metamorphose into a jellyfish, or a jellyfish may be produced through lateral budding. So just to summarize, Aurelia is a model system. It's also the textbook example of the jellyfish life cycle. You have a jellyfish that is the sexual stage, producing egg and sperm. And the zygote then develops into this free-swimming pannular larva that settles on the bottom, grows up into a polyp. The polyp produces baby medusae called ephyrae that then grow up into the jellyfish. So you can think of this transition from polyp to jellyfish as a rather radical metamorphosis, and you're familiar with other examples in the animal kingdom. For instance, in insects, the transition from a caterpillar to butterfly, or in amphibians going from a tadpole to a frog. And interestingly, there was a recent paper that actually suggested that the transition from polyp to jellyfish is actually regulated, at least in part, by the shared genetic toolkit, the same toolkit that insects and amphibians use. So this is quite a fascinating example, I think, of recycling in evolution. So this example I just showed you of Aurelia is part of what we call true jellies, or Scyphozoa, which is, again, part of this bigger group called Cnidaria. Cnidarians are jellyfish, hydroids, and corals. And without going into too much detail, I'm focusing mainly on this group called Hydrozoa, the group that contains hydroids and hydromedusae, two main clades, one hydroidolina, which are labeled polyp-dominant. Here, the medusa was lost multiple times independently or highly reduced. And my current work focuses mainly on Trachelina, a group in which the polyp was actually lost several times independently, and you could think of the jellyfish being dominant in this clade. So this is an rDNA phylogeny I reconstructed to study the pattern of life cycle evolution in Trachelina. The most likely scenario is that the last common ancestor of this clade still had a polyp and a jellyfish stage, which is still the case in this group called the Limnomedusae. You have a polyp. The polyp produces medusae, and all these, what they all share in common is they all live either in freshwater, estuaries, or shallow water habitats, so somewhere where you actually can find substrate for the polyp to grow on. And then interestingly, the polyp was lost down here in Limnomedusae and in this larger clade comprising Narcomedusae, Trachemedusae, and Actinolida. In that group, generally, the jellyfish develops straight from a free-swimming larval stage. And what they all share in common is they do all live in the open ocean. Some of them live in the deep sea. So in a habitat where there's nothing below you other than several thousand feet of water and then some muddy bottom. But you may have spotted in the annotation of my tree that actually we seem to have evidence of the re-evolution of a polyp-like stage in Narcomedusae. These are pictures that Karen Osborne took on a recent cruise out of in California. This is a little stolon. Here's where it used to attach. It's in the process of budding off a little baby medusa. And there's a second nubbin that looks like it's going to develop into another medusa. And so all these Narcomedusae still live in the open ocean, but they're polyp-like stages. They evolve polyp-like stages that parasitize either other jellyfish or, in this case, this pelagic polychaete. So this little stage, this stolon, grows in the parapodia of this pelagic worm. It buds off little medusae, and I'm not sure if you can see it in the back, but essentially the whole coelomic cavity is full of little baby medusae, or baby jellyfish. We don't know yet how they get out of the worm in the end, but they grow up into something like this, the jellyfish conina. Presumably the worm dies in the process. So this is my work on studying the pattern of life cycle evolution, but we also have this guy in culture called the Craspedacusta, which is part of Limnomedusae. This is the polyp we have in culture. Every so often, it buds a little medusa off the side. This is the baby medusa that then grows up into the adult jellyfish. And we would like to use RNA sequencing to determine whether or not the same suite of genes is involved in this transition as it is in the now relatively well-studied model system, Aurelia. So this will be a nice comparison. Morphologically, this transition is quite different. Rather than budding medusae off the oral end, they're budded off from the side, and so it'll be interesting to compare, see is it the same mechanism that is driving this. And so none of this would actually be possible without having like a group of enthusiastic colleagues, which I would like to thank. Don't have the time to thank them by name all, but yeah, thanks guys, it's fun to work with you, and with that, I'm ready to take my question. The question is, are there other jellyfish where we have examples where the polyp stage is a parasite? So there are several different ones described within this group, Narcomedusae. They all seem to cluster together, though. There's some really weird things that we now think are that are part of Cnidaria because they have nematocysts, and that's where some phylogenomic data seem to place them, called myxozoans. They're exclusively parasites, but they don't have a jellyfish stage anymore. You know how parasites are often just weird, they're highly derived, they look nothing like any of the other things anymore. And traditionally, there's another group called Polypodium that used to be grouped with the Narcomedusae simply because they have a parasitic stage that lives in the eggs of sturgeon. Hard to tell whether or not I would call that a polyp, but it does clone itself. So one thing that polyps do is they clone themselves. But now we place them outside of this larger clade. But they're sturgeon parasites. So there are actually several examples of cnidarians, or things that belong to the jellyfish clade that now are parasitic. Thank you.
Bastian, I have a question for you. You had a fairly robust phylogeny there. What's the taxon sampling like? Are there any species that you would really like to get but you don't have?
This one's okay. I think we have we actually have all of the genera right now, as far as I remember. I don't know what the exact count is. And since a lot of these things live in the deep sea, how many species there really are is not entirely clear, and oftentimes you have these species complexes. So that's obviously something that goes in that as well. Still trying to figure out which species are there, but so at the genus level, we have pretty much everything. And the whole group right now, there are around 90 or 100 described species as far as I recall.
And do we have all the tissue here, or do you collaborate with other institutions?
One reason why it's really nice to work here right now is that we have all the tissues and actually DNA extracts already banked here before I even started. But we still collaborate with other institutions that send us new material.
Thanks.